Hi, everyone. I am Brent, and I am happy to tell you about some of the stuff we've been doing in mechanobiology, starting with some of the tension sensor stuff we designed, and kind of moving into our latest goal is trying to develop assays that will tell us how force affects function of proteins in cells. And so I always like to start at the very beginning. I probably don't have to do this for most of this group, but I will do it quickly anyway. And so what we study is mechanobiology. And so a great part is, well, what is that? And I would say mechanobiology is defined as how various mechanical cues, such as cell contractility or shear stress, often which are described in forms of mathematical equations and things like that, get transduced into essentially something that affects biochemical signaling networks. And so that is what I have a depiction here on the other side is this kind of a simple network for how Rho and RAC, which are two of the main regulators of actin myosin, how those signal. So really we're looking at how these mechanical cues get converted into something that affects biochemical signaling networks. And so the next question you should ask is why should you care? And this is probably another answer that I don't have to spend too much time on here, but me and others in this room would probably argue a lot of the disease states that we don't have great treatments for have a mechanical effect. And so what do I mean by that? Well, cancer is a great example. So cancer is still driven by mutations, but if you look at the cancer microenvironment, you can see lots of mechanical effects, like cancer responds to the stiffness of the matrix around it, which can be changed by cross-linking, which is done by LOX. As cells grow, they push against their environment and generate stresses. And so this isn't by any means unique to cancer. You can also look at a lot of other diseases that don't have great treatments, and they have a similar mechanical kind of effect. I'm not going to go through these too much detail, but atherosclerosis is a disease with global risk. It's a leading killer in the developed world. It has global risk factors like your blood pressure, but it still happens particularly at places in the vasculature where it branches or bifurcates. And that's been tracked down to shear stresses being different there. Asthma is essentially driven by airway smooth muscle cells being too contractile and closing the airway too strongly. And essentially, muscular dystrophy is a particularly devastating disease, but it's often due to essentially muscle cells lacking a key protein and not being able to survive their own contractility. And so that's why it's known as a muscle wasting disorder. There's another one that I won't belabor, but it's called subconjunctive hemorrhage, which is not a disease. But if anyone, I don't know how big their screens are, I have one in my eye. That's why my eye is bleeding. That's often due to essentially high blood pressure in your vessels, but I'm completely fine. And if anyone notices my eye, there's no need to worry about it. So that's kind of the high level view, mainly because I'm in a biomedical engineering department and I do have to talk about disease. But what I'm trying to get at is there's probably hopefully some sort of commonalities underlying all these disease states. And so what we think that is called what's called mechanosensitive processes. And this can be broken down into a bunch of steps that essentially explain how mechanical forces can be detected by biochemical signaling networks at least in concept. And this process can be broken down into four steps: mechanotransmission, mechanosensing, mechanotransduction, and mechanoresponse. I know also probably many of you have heard these terms before. I have discovered that people tend to use them differently, so I thought I'd just take a moment and define them for how I use them. So mechanotransmission is the ability of a protein to kind of transmit force that's generated at one point, say, to another. This is a cartoon of an actin filament being pulled on by some myosin that's not depicted, and that force is being shunted through a protein that indirectly binds to integrins that can take it out to the ECM. And so where the transduction part starts comes in is the idea that proteins can change shape. And so this is well known in terms of post-translational modifications or structure functions from biochemistry. But probably about 25, 30 years ago, what we figured out is that proteins can also deform in response to mechanical loads as well. And that's what's depicted here in this mechanical sensing panel is we have a protein that's been pulled on sufficiently that the middle strap region has essentially changed conformation and exposing a binding site, which is shown in orange. And that's very typical of something that's supposed to happen in mechanosensing. Now, for this to have consequences, something has to happen, or biochemical consequences. And usually that is the act of some protein coming out of the cytosol and binding to that exposed binding site. And that is called mechanotransduction. That is the event where the mechanical stimuli has been essentially transferred into something that's biochemically consequential, and that's called a protein-protein interaction. And then what we talk about a lot is presumably downstream of that interaction, there's an activation of the signaling pathway. That is called mechanoresponse, and that can be not necessarily mechanosensitive in and of itself, but that drives the signaling that we usually think is causing diseases or cell migration or whatever process that you might be interested in studying. So the strengths of molecular mechanobiology are really the ends. We know quite a bit about cells and protein mechanics. As Colin mentioned, I've been studying cell mechanics since 2001. So we have some good ideas about how cells to form, how proteins to form. And because of the efforts of cell biologists and biochemists, we have a lot of idea about what these signaling pathways are at the end. What we don't know much about is this part in the middle where we don't really know. We've got this conceptual idea of how it works, but what proteins are being loaded? How are they loaded? Where are they loaded? How much do they have to be loaded to enable this cryptic domain formation or exposure? How long are they loaded? And if they are loaded, what proteins are forming these new complexes? This is absolutely critical information we need if we want to start to affect mechanobiology. The idea would be is we can go in and maybe if we can stop this complex from happening, we can stop the downstream signaling and stop the cellular response. And so that's really the motivation for kind of diving into the detail that we do, is that we really want to understand the mediators of mechanosensitivity. I mentioned it being an important disease. It's very important in physiology as well. Cell migration, wound healing, immunity all have key mechanosensitive aspects. And my particular dream, I didn't want to put goal, it's more of a hope, is that I did mention there's all these mechanosensitive diseases that we don't have great therapies for. Well, one hypothesis is that those diseases are essentially driven by when um, these forces in the cell become pathological or too high. So that means they can become pathologically high. If they pop something open, they've now formed a protein complex that's not supposed to be there. So then that protein complex is unique to the pathological context. So that's kind of an ideal drug target in a way, in that there isn't an endogenous pathway or a physiological pathway that we have to worry about, because it doesn't form unhealthy cells. So that is I won't even say 50,000 foot, it's the 500,000 foot goal of all this research is, can we start identifying these mechanosensitive protein complexes and maybe someday prevent their formation? And so how we're going to do that to start is we're going to design a lot of in cellulo, meaning I know uh, in vitro and in vivo are get confusing. In vivo, some people mean in cells. Everyone in BME means in mice. So I know it's controversial, but as I started using the term in cellulo, that means essentially we want to know what's happening inside, presume a living cell. And so I was very excited about this talk, and I probably have way too much here. I've identified parts of the talk where I can skip, but I realized this is probably the first time and a good venue where I could kind of enumerate my entire vision for the lab. So I kind of got mechanobiology tapas for you. So I'm going to jump around and just kind of hit some highlights with kind of emphasizing the main goal of kind of our research thrust. And so what's maybe unique about mechanobiology in terms of some even traditional mechanics or traditional biochemistry is it's a relatively new field. We didn't teach too many mechanics people about biochemistry, and we didn't teach too many biochemists about mechanics. So one of the main things, and that's what this center is so wonderful for, is a key limitation of mechanobiology is developing the necessary mechanotechnology. And so I'll talk a little bit about our version of that. And so a lot of what we've seen is beautiful DNA-based nanosense tension sensors, which I am jealous of the Cellulo noise and how wonderful they are. We do a slightly different version because we want to start understanding a little bit more what's going inside a cell. So I will talk to you about how to design a genetically encoded intercellular molecular tension sensor, or MTS for short. We've had a kind of revelation a little bit, that everyone uses GFPs, or essentially glyphosate fluorescent proteins in these sensors. We'll talk a little bit about this. GFPs weren't evolutionarily designed to be loaded, so there could be some consequences for that. And then I'll tell you about this. Most all our work is done on this, what I consider a archetypical mechanosensitive protein, vinculin. I'll explain what that is, and it's essentially a protein that we know what it does biochemically. We have an understanding of how it behaves mechanically, so it works very well for kind of developing technologies. And so What we'll focus on is this approach development. That's the title of my talk. And we're going to talk about two functions of proteins. Um, if a protein, as I even described in the beginning there, the green protein links the F-actin to essentially the integrin. So that's a mechanical linking protein. And a major function of proteins, of linker proteins, is to maintain that connection under load. For those of you who don't know, I'm basically getting at catch bonding, and I don't know if Cheng Zhu is here, but he's obviously one of the founding fathers of that field. Then the other thing we'll talk about, another function is this force sensitive protein complex formation. And I'll give you two ways that we can do that. One is with imaging that kind of enables hypothesis testing. Does this protein form a force sensitive construct or force sensitive complex? And I'll also tell you about some very new data where we've found a way to do this in an unbiased manner where we can ask if proteins form tension sensitive protein complexes. So this will also be the running outline for the talk. So let's get into a little bit of how we build these tension sensors. And so if you're going to study something on the molecular level, you have to have a technology that's sensitive to essentially nanometers, nanometers and piconewtons. And so just like in the DNA sensors that we've heard a lot about in this seminar series before, almost all of our technology is based on FRET. And so FRET is a non-radiative energy transfer between two fluorophores. In the simplest terms, and I know this isn't quite physically, but it works out well color-wise, is you shine in some blue light and that connects to what's called a donor fluorophore, which can also be a fluorescent protein. That protein can then become excited and it can pass energy to a neighboring molecule. And if that one is a diff proper energy level, and of course, energy level corresponds to wavelength of light, it will emit light. And then, but this fluorophore is different, it will emit a different color light. And so this means you can essentially shine in blue light and get yellow light out. And that's something that's very easy to detect on modern day microscopes. What makes this very, very special is this phenomenon is exquisitely distance sensitive. This energy transfer only can happen, at least for fluorescent proteins, on length scales of about, say, 4 to 10 nanometers for typical fluorophores. So this is a great readout for us to be able to see how two dyes would move in space. And so everything I just told you about was distance. And what we're going to be doing is a tension sensor. I will do my best not to call them force sensors. While there are some force sensors, force is a vector. And so force, that means you need an amplitude and a direction. Our sensors have no direct vector information, no directional information, so we just call it a tension. And so what all these are essentially two fluorophores that can fret linked by a spring that we know the mechanical properties of. And so the entire game that we'll talk about, and this is why I probably mentioned I talked about statistical physics, is that we need to know what the mechanical properties of that linker is. So it needs to be calibrated. And then if you have a calibration, you can take this thing that had a distance sensitivity to it and then enable it to have a force sensitivity. The way you do that is you essentially put handles onto the tension sensor. That handles will be the embedding protein that you're going to study. If that protein gets loaded, then the spring will extend, the fluorophores will move apart, and the fret will drop because they moved apart. And so you'll get curves like this where you can have very high fret efficiency, so lots of yellow light coming out, that drops as a function of applied load. And so this is kind of as one of the first calibrated tension sensors. Did this in the lab in Martin Schwartz when I was a postdoc with Karsten Grashoff. And so this is one of the first times that we could visualize calibrated loads in specific proteins inside cells. So to jump ahead, this this technology has been widely used. So this is kind of one of the gold standards for measuring proteins inside cells for the forces they experience. So today I will be talking to you primarily about vinculin. I just wanted to take a second to say that this technology is not unique to vinculin. It's been placed in a lot of other proteins, such as cell-cell receptors, cell-cell adhesion molecules, I apologies to the Springer lab. I realized we forgot to mark the integrin sensor that links focal adhesions to the ECM. These are some focal adhesion proteins, actin, skeleton. All of these proteins have been altered to incorporate this tension sensitive module and report the loads they experience in some context. And so since this is the technology center, I thought I might spend a little bit of time about how we build and understand these things. And so this is the concept I just showed you. If protein gets loaded, the spring will extend and the fret drops. And so there's a couple key parameters that you have to consider. The first thing you have to consider is what's called the Forrester distance. I'm not going to spend too much time on this, but that is just a measure of how good a given fret pair is. A bigger Forrester distance means a given fret pair can mediate fret under a longer distance. What we're going to spend most of our time talking about is this spring. And there's two things that you can kind of control. You control the length of the spring and you control the stiffness of the spring. And so these don't have to be springs. They can be structured domains. For instance, Carsten Grassoff has done a lot of work with structured domains and more digital-like sensors. Our particular group's idea was that can we use an unstructured polypeptide? And then that's because this is well understood in terms of statistical physics, and we can make models that how this will kind of behave a priori. Because the dirty little secret of that original tension sensor is we made 50 versions. We made 50 versions, never told anyone about the other 49, and published one in that initial paper. So I was like, can we do a little bit better than that? And so the reason I like this is there's a very simple kind of standard entree into how you can do this, and that is essentially the very basics of polymer physics. And so if you have a polymer at the molecular scale, what happens is, of course, it gets buffeted about by Brownian molecules, or by Brownian motion or water molecules. And so it turns out how straight the chain is an indication of how stiff it is. And basically that means how well it can resist that Brownian motion. So if you have an infinitely stiff polymer, essentially it looks like a rigid rod. And this is something that we're used to on the macroscopic level. And the other extreme, if you have a very, very, very soft polymer or flexible chain, it can't really resist this Brownian motion at all. And essentially it goes up into a random coil. And so this gives us two length scales that we have to consider. One is that what's called the contour length of the protein. That's how long it is if we pull it all the way out. And the other one is the persistence length. And so the persistence length is a measure of how long, if you move along the filament, you can estimate the direction it moves in. So here the persistence length of this rigid rod would be infinite, and the persistence length of this flexible chain is very small. And so it turns out, if you're a good physicist or you're a good engineer, you love dimensionless groups. And so there's a dimensionless group, which is the ratio of the persistence length to the contour length that defines whether something's flexible, rigid, or somewhere in the middle called a semi-flexible filament. And so now I did mention that these are all essentially buffeted about by Brownian motion, so the ends aren't still, they move. And so that means you have to think about the end-to-end distance as a fluctuating entity, and that means it's easiest to think about as a probability distribution function. Okay, if you're confused by probability distribution functions, just think about a Gaussian distribution of grades, and it's the same idea. Okay, and so if we're going to make a spring to make one of these sensors, it's very easy for us to control the contour length because we make it in DNA, we specify the number of amino acids. So we have direct control over the length of whatever deformable element we're going to use. The thing that changes is the stiffness of that linker. And that is going to be set by the composition of the amino acids that we use. But polymer physics tells us that all we need to know is the ratio of that contour length to persistence length. And then we can, this is basically a variety of different approaches for predicting that probability distribution where this is the only input. The LC to LP parameter persistence length is the only input, and that goes from something, if it's 100, that's essentially like a flexible chain, and that should look a lot like a Gaussian to you, like a random coil. And that proceeds all the way down to almost something that starts to look like a rigid rod, which would be a delta function at 1 on this plot. And so that means we can actually use this if we set the contour length to a constant. We can use this paradigm to calculate the persistence length of any given filament. And so that means we can make something that's got a length. We can stick two fluorophores on it, and then we can use fret as a readout because that will tell us the distance of the two fluorophores. Fret occurs at the center of the chromophores. That's the relevant distance. Sorry, that's not where it occurs. That's the relevant distance for fret. So you have to do something to account for the finite size of the fluorescent proteins. It's all at the molecular scale, so everything's rotating. So you do have to do something to handle the rotation of the fluorophores. But if you do that, you can have basically a way to measure fret efficiency, and determine the persistence length. And so we did that for a bunch of linkers. And so we were very excited. We took linkers and we made them different lengths and we measured the fret efficiency and we got data that lied on a nice curve. And then we could take that model that we just derived and then we can measure the persistence length. So that means with this assay, we can get everything we need to know to understand how an unstructured polypeptide functions mechanically. And that is, I think, what something for people forget. Most of the time when we talk about a spring or a polymer, we talk about a force extension curve, which is shown here. This is the famous warm light chain extension curve. And if you know what the probability distribution function is of your filament, you can directly calculate this. That's just what's shown here. All you need to know is the distribution of essentially the angles the chain can run through. And that same exact information that dictates the probability distribution dictates the force extension curve. So that is why I talk so much about this, because with this persistence length, we can essentially make springs of known mechanical properties. And that also means, or there's one, if those are related, we can take this little procedure that we created to measuring the persistence length, and all we have to do is put it in a for loop. Because we can just propagate how that probability distribution changes as a function of force. And then that is exactly how we determined the mechanical sensitivity of these fret-based linkers that we talked about. And so that is what we can do. And if you're a good engineer and you have a model and it's got a big parameter, you run it over and over again. And so what you can see here is essentially this is the persistence length. This is the number of amino acids. And then what this point is every single one of these dots is a simulation of a particular unstructured polypeptide in a fret-based sensor. And so one thing that you care about is when you extend it out, what's the change in fret? You want this to be high so you can have a sensor that you can measure well. The other thing that you might care about is where that force is happening. And so what we did is we took that range of the fret and found its mid fret change and found this midpoint, and we called that the target force. You can do a same exact operation with extension. And so I believe what we said is, and then this is also particular for a given force or distance. So I think when we did this, we made predictions about the behavior of a couple thousand different fret-based tension sensors. And so what this means is you have different proteins under different loads. You can make different sensors to measure those loads. And so that was how we think about it. That's how we make it. And I would argue that this is unique in that this enables rational design. If you have a protein and you have an idea what the force is, we can build you a sensor that is optimally sensitive. And so when I claimed victory, I love this paper. I was like, we solved it. And then a very well-trained former grad student of colleagues came and joined my lab. And he was like, dude, you said all this stuff. You show me you have linkers and you have one linker. You have one linker, you can make just one lane. Can't make you, so I can predict all of these, but you can't produce them. And so what he did, and one of the many things he did in the lab, was try and find a way to expand this persistence length we had accessible. And he had a pretty simple idea. He picked some common amino acids, some that give very soft filaments, some that give a little stiffer filaments in the polyproline, and also ones that kind of can control the hydrophilicity or how much it likes water or not of the amino acids. And then he also didn't want to do this in microscopy, so I found a way to do the same exact thing in flow cytometry. So very quickly, he was able to build a library of many linkers and measure their persistence length by doing the same exact thing we did before and making them different lengths, but by measuring it in flow cytometry. And so before we only had a linker that was a persistence length of 0.5, and he was able to expand that out to basically something that's almost a persistence length of 3. And so that may seem like a small difference, but if I show where that range is on these plots I showed you, we now can kind of span all the best part of the region where the force sensitivity and the fret sensitivity is changing for all these linkers. So this enabled kind of this vision of actually being able to rationally design a tension sensor for a given force to come true. So these linkers will be hopefully released soon and people can start using them to measure forces and even more different types of proteins. Okay, so that's statistical physics and all those things. And basically, what's cool is we used entropy to build a tool. This isn't based on enthalpy or anything like that or crystal structures. It's just an understanding of fluctuation of polymer filaments to make a functioning sensor. So next thing we have to think about is the fluorescent protein. And so this is new. And so this is not changing the force or distance. This is something else. You'll note in the sensor design that the fluorescent proteins are in line of the force. And so that means they can be subject to mechanical load. This is not typical how these proteins are used in nature or in engineered sensors. They're usually stuck at a termini so they can glow. And so after working for the sensor for a while, we got a little bit worried that, well, what is force doing to these things? And so, as we often do, we went and looked at kind of the physics literature. We went and looked at the single molecule physics literature. And there is this very nice paper from Matthias Reef where he showed that if you load a fluorescent protein long enough, it will eventually, here is it's loaded, and this is what's showing it's excited here, it's emitting photons, it's emitting photons, it's emitting photons, and eventually it stops. Okay, so force can make a protein stop working, and that's called mechanical switching. What's good is if you wait a while, you unload it for a little bit, and then you try and excite it again, and it'll come back. So this is reversible. Okay, and so there was a variety of reasons where you're interested in this. Is if we have mechanical switching, the calibration that we spent 10 years trying to do doesn't work. But there's also some opportunities here. Fluorescent proteins are proteins. And so if they have force sensitive functions, they can maybe potentially start to help us understand how other proteins or parts of proteins can have force sensitive functions. And so all we're kind of showing here is this change in function is due to a change in conformation, just like something might have happened in for one of these linker proteins we talked about before. So we thought it might be a new way to start talking about mechanosensing. And also, one of the things that's really come up in the DNA sensors is what is the loading duration of a protein or loading rate of a protein or kinetic phenomenon. So for our entropic linkers, all the changes are instantaneous. They're super fast. So if the duration of, if two sensors are loaded to the same amount, but loaded to a different duration, we can't tell. And so there is something known about how these things unfold, and it's basically a two-step process. Um, and then the idea is there's supposed to be an initial transition that's almost switch-like that happens at equilibrium at a given force. And after that, there's kind of a traditional kind of force-activated process, which can be styled by a Bell model that can be slow. So you have to kind of pop out one helix, and then you pop out a second helix, and that's what leads to the loss of function of the protein. So these dynamics can be much slower than what's going on in our entropic-based sensors. So we thought this might be a new window in which to study mechanobiology. And so we basically set up a kind of large team for us to go ahead and do this, and it's going to be almost all modeling. And what I discovered over the course of my career is we do lots of things. We write papers, and then we put a lot of them in the supplement, and then I end up showing like 2 figures of it in the talk, which I decided is kind of unfair to the students. And so I will show you who did the work, and I will show you all of the things they did, even if I don't show you all of the results. And so this was a large mix of modeling, kinetic Monte Carlo modeling, steered molecular dynamics, analytics to help us get a handle on what was going on here. And we started with the simplest possible model, where we just have a tension sensor that's loaded by some force by some time. So you can think about this as this little top hats. And we embedded in it 2 different fluorescent proteins that could undergo this mechanical switching as I described to you earlier, and we'll just note that these two do not have the same properties. There's different fluorophores, there's different structures. We didn't think it was appropriate to pick the same kind of conformational change kinetics for the different fluorescent proteins. And so this is what this ends up looking like is you have a tension sensor, and then you, instead of both the fluorophores working, you have a mixed population where some of the fluorophores, including both, can break because they're under too much load. And so what we did is, one, if a sensor is functional, we considered its force sensitivity to be set by that linker, just like I showed you before. And then there's going to be what's called extra fluorescence or extra fluorophores around that don't really contribute to FRET, but can contribute to optical signals because they are functional but lack a FRET partner. And so I am not going to explain it in detail, but we came up with a way to turn this kind of idea into representation of the optical signals that we get in our microscope. And then I'm going to generate for you what I call data fingerprints. Just what does this look like if this process is happening or not? And so how we're going to visualize this is a combination of two parameters. We'd like to call these ES histograms. The E is the FRET efficiency. That's the thing I already told you about. That is a measure of how much this fluorophore can pass energy to that. But there's going to be another parameter, which is called the stoichiometry. The stoichiometry is a measure of how many fluorophores are functional in a sensor. So this thing is functional. And so it's got two fluorophores, 1-to-1. That's defined as the 50% stoichiometry. There was already a formalism for describing this because many people use fluorescent proteins to study protein-protein interactions. But they do that in what's called a split sensor. So it's not one piece. And so they were interested in understanding what happens when you have extra fluorophore of one type or another. And it turns out if you have extra acceptor, the apparent FRET efficiency changes from this true FRET efficiency because this contaminates your optical signals. And if you have extra donor, you now get a correlation between the apparent stoichiometry and the apparent FRET efficiency. So essentially, extra donor will drop your apparent FRET efficiency, which can make sense because you have no acceptor to FRET with. Okay, so all we did was take their formalism, and instead of just mixing in free donor or free acceptor, we made it force sensitive so that as this thing gets loaded, we could account for the various fluorophores breaking. So what I'm showing you is the simulations of sensors loaded with a given force range or a given time over a range. And if you have no braking, the sensor looks exactly as we intended it. Basically, the stoichiometry stays at 0.5, and as you apply more and more force, the FRET drops. What I'm showing you here, for reasons that will become clear in a little bit, is that if you have large acceptor braking, as you load the protein for more or longer, now you'll see this apparent change in stoichiometry. And so what this means is this calibration doesn't work anymore. And the sensors are sensitive to both the amplitude and the duration of the load across the sensor, and they can't be separated. Okay. So what we are saying is this is not, you just need to know if this is happening. This doesn't break genetically encoded tension sensors. What it does is you need to know which mode you're in. If you aren't undergoing this mechanical switching, then you can use this sensor as intended. That's the mechanosensing mode. If you start to see this switching, that means one of your fluorophores is starting to fail. And now that doesn't mean the sensor is broken. It just means it can be used in different ways, potentially because this is an indication that that acceptor here has been loaded to such a degree it's no longer functioning. So that means any other protein domains that might be in the sensor that are weaker than that domain could also be opening. So what we're saying is this is now a new opportunity to start visualizing if mechanosensing or a protein popping open is happening indirectly. Okay, so that is how they use the sensors. That's how to make them. And we basically have generated a new information stream, which is the stoichiometry. And so what we're going to do now is put it in a physiological protein and actually use it to try and learn some biology. And so the protein that we're going to use is a protein called vinculin. So vinculin is a key adapter protein that exists between, say, force-rendering active machinery and integrins, which are transmembrane proteins that bind to ECM, and it's thought to be involved in force transmission there. This structure is a depiction of what's called a focal adhesion, which is how cells stick to their environment. Vinculin, if you look at all of the proteins in a focal adhesion, actually forms more connections with the other proteins in a focal adhesion than any other protein. So it seemed like, well, worthwhile for studying in terms of if it formed force-sensitive interactions. And it's also known to be very sensitive to the stiffness of the environment. This is some work by Val Weaver that shows that basically that showed that if you change the stiffness of the environment, you can change how vinculin is positioned and loaded inside a cell. Okay. It also is important for a lot of module functions. If you get rid of vinculin, the cell's traction generation will change, traction force generation will change. This is a very interesting result from one of our collaborators, Cindy Reiner King at Rice. Turns out if you make a little pore about the size of a cell, these things can be as a channel, and these can be found in something like in cancer microenvironments. Generally, the cells will do what's called super persistent migration. They just walk all the way along the end of that tunnel until they hit the end, and then they walk back. And that does not happen if you don't have vinculin. And this is this kind of this collagen contraction assay is kind of a classic model of how cells can work on tissues. The more contractile the cells are, the more that collagen gel will shrink. And so vinculin is important to many aspects of a cell's mechanics. So we took, to make the vinculin tension sensor, we took this tension sensor module with a spring based on spider silk that does behave just like an unstructured polypeptide, did a bunch of cloning, stuck that in the middle of vinculin, and now we have a very powerful tool that can tell us the localization of vinculin by exciting this acceptor, because acceptors can't fret, and the force across the vinculin by directly exciting the donor here, which is called, which is MTFP1, hence the T. So the way we do this is we get vinculin null MEFs. We basically use lentivirus to basically rescue these to endogenous expression levels, which we identify by sorting. And then we take the cells and we place them, and this is the joke, on their natural environment of fibronectin-coated glass. We do this so we can have good imaging, and because we're interested in some of these force-sensitive properties, which have been characterized in the glass. And so we can do that here. And then if you do everything right, it'll look like this. And then this is amenable to things like automated image analysis algorithms and things like that. And so if you do everything right these days, this is what it looks like. This is a cell expressing the tension sensor. This is the acceptor channel. So this is its measure of its local concentration. This is the fret signal coming off of those focal adhesions. And what you can see right away, this is color coded such that unloaded is probably this yellow-orange color. That's the unloaded fret efficiency for this linker. It's about 29% fret efficiency. And this is usually sometimes surprising to people. There are focal adhesions that have no loaded vinculin. Okay. It's not like every focal adhesion is bearing load. And then as you can see, these colors drop. That means there's more and more force, and we can see or tension across this protein. And that primarily happens in bigger focal adhesions towards the edge of the cell. That is the fret efficiency. This is the stoichiometry thing I mentioned before. And here we can see that there's not very much variation in the stoichiometry. It's very close to 0.5. And then this is a check, and you can use this sensor in this cell to calculate forces. What took us a while to see this is if you look about 10 to 15% of our data, the fret will look exactly the same, but basically the stoichiometric signal changes. So this means we do have this mechanical switching going on in the sensor. The acceptor is breaking, and that's why I showed you this plot kind of before. So then that means in this particular cell, the forces are such that we are rupturing that acceptor. And then this is now what we're arguing could be indicative of some sort of kind of mechanosensing process going on in the vinculin. Okay, so I'm going to skip some stuff, but I'm going to do these last two parts. And so then now that we have sensors and we understand how they work, we can start talking about how they force on a protein that changes its function. And as you can see, the force function I want to talk about is a protein acting as a linker. And so all of these structures link one structure to another, and almost all of them are linked to a force generating actin cytoskeleton. And then sometimes there's multiple linkages that link to a force resisting structure, such as the ECM. So cells want to maintain these linkages, and so that is a function of linking proteins. And one thing you may have heard of, if you've talked about this, is essentially the idea of a slip bond versus a catch bond. And that is, if you pull on something, it's got two choices. It can either unbind faster, or, and that's what you're used to in your everyday life, or it can do this relatively unique thing called a catch slip bond. We are not as used to this in our everyday lives, but this happens in a lot of adhesion molecules. Essentially, is that as you pull on this linkage, the bond lives longer. If you didn't like the mathematical descriptions, here are two cartoon stories. If it's slip bond, that's what you're used to with tape, is that you pull on it and then that basically will unbind faster. Here is a depiction of a macro scale example of a catch bond, which is essentially this finger trap toy that you pull on it and the lifetime lives longer. And of course, one of the very first seminar series we had here was Cheng Zhu, and Cheng Zhu has kind of been the founding father of this field and has mapped out, I think he said 40 of the 60 catch bonds or something like that, which is amazing. And so the reason we care about it is many of these catch bonds are essentially in complexes of integrins and fibronectin or actin and myosin. So all of these mechanical linkages that hold tissue together have this catch slip behavior. So I would argue a major thing we need to do is figure out how these work. And so this is a data from Alex Dunn, but what's very interesting is it shows that vinculin is a directional catch bond. You can pull on it one way and it forms a strong catch, and you can pull on it another way, it forms a weak catch. And so we thought this was an interesting behavior we could leverage to answer some questions about catch bonds. Essentially, how they're working, and can we manipulate them and figure out what they're doing inside cells? And so to do this, we teamed up with Sharon Campbell at UNC and two of her postdocs, Venkata and Mohammed, who did a great job. Venkata did a lot of simulations, and Mohammed's more of a traditional biochemist. Um, and so here is the fundamental observation we had. So this is an actin, two actin protomers that have been polymerized, and this is vinculin's tail domain. And so here what they're doing is applying a force this way, and here what they're doing is applying a force that way, okay? And what you can see is when we apply this force, this vinculin tail domain is never unbinding, and when we apply the force this direction, it is. And that recapitulates this directional catch bond. And so what the idea was to figure out how this works is we could look at this interface in simulation and ask the number of connections that vinculin was forming, and we pulled one way versus another, okay? And so generally what catch bonds are was when you pull on some direction, you make a bunch of interactions that stabilize the bond. And so this was true here. I'm going to skip it. I understand there's other types of interactions. I'm just going to focus on the hydrogen bonding. And I understand this plot is complicated, but what it shows is the interaction between amino acids and vinculin and amino acids on the actin protomer. And everything that's a star is an interaction that occurs in the unbinding and unloaded interface between vinculin and actin. And then what you're seeing is they basically increase the force, and the bigger the ball gets, essentially the longer that bond survived. And so this is all to show you that we identified two amino acids that basically have catch-like behavior only in the loaded state because they aren't marked by a triangle. So what that means is we can go in and mutate them and see if we affect vinculin's ability to catch bond. And so that is exactly what we did with a tension sensor. We went in and put in all these mutations. And then we were very excited to see that when we did this, we progressively unloaded the catch bond, unloaded the vinculin, but not completely unloaded it. This is a different mutation here that completely unloads the vinculin. So this is exactly what you'd expect from a catch bond. And so we were very excited. We can also see that this presence of this catch bond basically was required for this breaking of the acceptor. So without it, the tension functions normally, the sensor functions as a emission sensor, but with it, we can start to see this breaking of the fluorophore. So we're moving from sensing mode to transmission mode based on a biological change in function of the protein. Okay, I'm going to skip that. That's just another way we showed that we're vectored the catch bonding, but now that we can ask for the first time that I know of, if we get rid of a catch bond, how does the cell's behavior change? And so Julie was a student in my lab, and she did all those assays on the right, And then when we got rid of catch bonding, all of those things changed. Without the ability to form catch-gone, vinculin couldn't form, focal adhesions couldn't mature. You couldn't get the proper co-organization between stress fibers and focal adhesions. And the one I'm going to show you is that cells couldn't undergo directed migration anymore. So this is a very simple, very classical assay. What you do is you take a Boyden chamber, and you coat it with fibronectin on the bottom. And this is a measure of haptotaxis. If you have just fibronectin down here, you get a gradient. So if the cells can sense and crawl along that gradient of fibronectin, and what you're seeing here is that normal cells can do that quite well. If you don't put any fibronectin, no cells come through. But if a cell can't form this catch bond, the migration is greatly perturbed. So that is one of the things we're most proud of. We now have assays for being able to tell when when a protein's catch bonding or when it's not, and how it affects cell behavior. In the last couple minutes here, what I'm going to do is talk about this last one where we tried to build technology to start studying force-sensitive protein-protein interactions. And so this was inspired by the literature. This has been done a lot for a specific set of proteins. What's shown here is you can take a protein called talin, and if you pull on it, you will expose cryptic domains in these various alpha helical bundles, and that enables vinculin binding. Here's another pretty famous one. You can take the acting cytoskeletal cross-linker filament, and if you shear that network, what will happen is when the filament's unloaded, it'll bind a fill gap, which is just a row regulatory protein. If you pull on the filament, it pops off. But basically, what doesn't just nothing happen, then basically what happens is another one, this one is beta-7 integrin, can come in and bind. Also, if people are in this field, you've probably heard a lot of the limb domains. There are ideas that limb domains can bind to essentially cracked or deformed actin filaments, and those are all examples of this force-sensitive complex formation. So vinculin does unfold. This is in a preprint from Ben Galt's lab, and so we're wondering if vinculin can do the same type of thing. We have two different ways to do this. One is essentially with imaging. And so this is published in a developmental cell paper from 2023. And so what all this is it's based on the idea of co-localization. So if you make one protein green and one protein red, if those two intensities correlate, then it's likely those proteins maybe have some sort of interaction. We just updated this idea. Instead of having two proteins, we had the tension sensor and a protein of choice, and the idea is the same. If these two signals correlate, there might be an interaction. So this was published, and I will say we swept a whole bunch of focal adhesion proteins. We can find some that care about vinculin tension. We can do some that don't. Because this is published, I'll just let this look it up. But the way to think about this is you can do you can do hypothesis testing. The one I want to tell you about is this one, where to do this, what we do is we use vinculin that can be loaded, or vinculin that has that point mutation so it can't be loaded anymore. And so the idea was, if there's a technology called TurboID, which will generate a biotin that can bind to any surface exposed lysines. So this thing gets activated and binds what's nearby. So it's called it basically shows proximal proteins. So we took this thing and we put it on either vinculin that could be loaded or vinculin that can't be loaded. So that means we would identify, hypothesis was any procohesion protein, proximal protein that required load to be near vinculin would show up in this pool, but not show up when we used this particular mutated construct. This has all been done by Megan. And so we basically took the vinculin that could be loaded, the vinculin that couldn't be loaded, put this turbo ID on it, rescued our vinculin null cells again, did a cell lysis and a biotin pull down, and sent all that for mass spec. And so this is just to show you that this data looks as very high quality. We basically got all of the members of the core focal adhesion back from these two constructs when combined. But what got very interesting was when we looked at what proteins were enriched on vinculin that couldn't be loaded, or enriched by near vinculin that could be loaded. And so that doesn't make any sense. So then this is what if you look at this is probably what you're interested in. There's all the proteins we saw. And I brought up some of these examples from before. There's this filament switch, something that's enriched near vinculin when it's not loaded. Fill-Gap's other name is RGAP24. So when vinculin's not loaded, we saw components that are associated with the lack of loading of filament. And then often over here, when vinculin does get loaded, it can form new interactions. Some of those are fibulin or McFillin, or FBML1, or CSRP1. All these things with an FM after it are essentially things that interact with filament in a force positive manner. So by manipulating this vinculin switch, we seem to get the interactors of this established filament switch. So that made us very excited. If those of you in the field, we also got a bunch of YAP regulators, which is a main regulator of how cells respond to stiffness. And we also got a lot of limb domain encoding proteins, but we got the ones that don't bind this actin cracking thing, that don't bind these these actin filaments. So it suggests we got a new type of interaction with limbs, and we were very excited to follow that up. I'm going to skip that. Okay. So I'm sorry I put too much in this, but I was super excited to come and talk because I thought this is the first time I could kind of show the idea of what my lab has been doing. We've been trying to start basically improving technologies to understand mechanotransmission, understand mechanosensing, and understand mechanotransduction. And I've shown you that over the past 13 years, we have done just that. We started by making these tension sensors that could tell us about force transmission. We've brought in technology about how to start talking about catch bonding, which determines how long and what amplitude these the forces these proteins experience are. We've brought some new tech for starting to tell us when we think there might be conformational changes in the protein. And we've also finally moved into being able to tell and discover force-sensitive protein-protein interactions, which we think are the basis of this mechanosensitive disease. So this is important because this is all tech development. I'm not entirely sure vinculin is super important in every mechanosensitive disease. I'm not saying that. But what we've done is everything we've done is all it requires is a functioning sensor. And it works a little bit better if you have a point mutation that can unload the sensor. So all of the approaches we have created can be used for any protein that has a sensor. And so we have these ones coming up on the docket, this NCAD and ESRIN sensors. So we think this is a general procedure that we'll be able to use to really kind of push forward the mechanistic basic of mechanotransduction. So I am sorry I went so long. I did intend to leave time for questions. I was just too excited. My lab really likes Halloween, and I am excited that we can do that again. I think I thanked everybody who did all the work along the way. They're all boarded here. And I would like to thank my funding from the NIH and the NSF. And if there's time, again, I'm sorry, I am happy to take any questions. Thank you.
That was great, Brent. Awesome.
Sorry if I overloaded it. I was just so excited. I was like, I can finally say the whole thing.
I don't know. I think the audience was like a catch bond. You can't overload it.
Nice.
All right. I'll let if you have questions, please you could put them in the chat. You could raise your hand and unmute and ask questions. That was really great. I'm going to ask a question since I don't see any other questions right now.
Mhm.
Um, so I was I have so many questions, but the GFP mechano switching.
Mhm.
Do you think that requires growing a cell on glass slide? Do you think it's an
Um,
artifact of putting cells on hard surfaces?
so I don't think so, yes, but I don't necessarily think it's an artifact. So this is something I didn't show, but if you do put these things on softer gels, the mechanical switching will go away. And if you get rid of the catch bond, it'll go away. I think it's kind of the way I think about it is and mechanosensitive disease is extra stiff. When we're putting cells on glass, we've been studying the pathological state. Does that
Right.
is my hope, at least. So that's how I spin it, if you will. But yes, it is stiff and sensitive, and these things do seem enriched on stiffer matrices.
Yes. I see.
Hi, Brent, can you guys hear me?
Yeah. Yeah.
Yeah, this is Jim Sellers. I can't figure out how to raise my hand. I'm just going to be rude and jump in.
Go ahead.
Also, this is my first one of these seminars that I've tuned into, and they're very useful. If they're preserved online, I may go back and look at a few of the other ones. But I have two questions. One of them is sort of a follow up or an extension of what was just asked.
Mm-hmm.
You know, a lot of people are looking at cells in 3D matrices now.
Mm-hmm.
Have you tried to do any of that to see whether you can get signals, or is it too far away from the turf field?
So, well, we don't use turf, but yeah, we have not done. It took us like 7 years to figure out how to do this on gels. So we haven't done much in 3D matrices. Others have. It turns out like there's all different nuances in how you do the fret, and if you want to get the fret efficiency and forces, which does help the interpretability, it makes it harder. You can use it in a mode where you just kind of get relative forces. And that has been done in 3D and in like embryos and things like that. Is that so? It's not that the forces go away, but they do get smaller. Is that
Yeah. Okay. So then the next question is reflecting my interest. For those that don't know me, I'm a myosin guy, and the question is, if you throw blebbistatin in, do you get force without blebbistatin, or do you always, I mean, without myosin, or do you always need myosin activity? Is it the only thing generating force on the actin, or are there other mechanisms?
there are other mechanisms, definitely. So we can basically if we make different types of vinculin, we can see one sub by mutation, we can see some interactions with vinculin. Essentially, if you kind of block its ability to bind to talin, it can be loaded in an F-actin polymerization type manner, but not a myosin or at least by Y compound type manner. The normal state, if it strongly interacts with talin, then the loads do predominantly seem to come from kind of rock or myosin-based mechanisms, but do not care as much about actin polymerization. So it does. I might think some of the being on glass and being on stiffer surfaces seems to bias it towards this completely myosin dependent or more dominant to the myosin dependent. But as you go to other environments or other things, you can start to see some of the actin polymerization based loading.
Thank
Then
you.
Great. I think the next person that raised their hand was Bronson, and then Chang, and then Viola.
All right. Thank you very much for the talk, Dr. Hoffman. I wanted to ask a little bit about the data acquisition.
mm-hmm.
So I'm assuming you've done all ratiometric measurements, right? So you get the MTFP, the venous, the MTFP quenched, right? Like all that.
So we do what's called this gets okay. So that's a great question, and I did
Yeah,
skip all that for the sake of time. We do what's
sure.
called three picture imaging FRET. We do
Yeah.
donor in, donor out, donor in, acceptor out, acceptor
Yeah.
in, and acceptor out, and then we correct for bleed throughs. And then we
Hmm.
do other calibrations that other groups don't do, where because we can make all these modules. It turns out if you have constructs that are of unknown but different fret, you can use that to calibrate your microscope. And so I
Okay.
did say fret efficiency. We did measure fret efficiency. That is the distance
Mm.
sensitive one. It is not the more, which I think you're getting at, which is not the more common fret index. And so it is
Yeah.
not true that you have to do FLIM to do fret efficiency.
Oh well, I was going
Yeah,
to actually that was
yeah,
sort of got to my second part
yeah.
is what are your thoughts on using these types of sensors with a FLIM-based system.
So
Uh,
these types of sensors with the unstructured polypeptides are probably
yeah.
horrible for phlegm because
Okay.
they have a very, very, very high number of lifetimes. Essentially, the lifetime is continuously distributed with
Hmm.
the force. And so that's bad for phlegm. There are
Okay.
other types of sensors, like the DNA sensors we've seen before, or some made by Karsten Grassoff, and Khaled has a new one, I believe, as well, that are more digital, that they kind of pop open in a given force. So then now you have kind of an unloaded state and a loaded state, and now you have fewer lifetimes that you have to do, and then you can do phlegm much easier with those.
Okay.
But this tie it back around, if you have mechanical quenching or mechanical switching in your phlegm acceptor, you will not
Hmm.
see it in phlegm.
Okay.
And so
Yeah.
there is,
Yeah.
yeah, yeah,
Okay.
yeah. Does that, so there is reasons to do both. Does that, yeah,
Okay. Okay. Thank you. No, I appreciate that.
sure.
All right, Chang.
Hi, Brent. Great seminar.
Oh,
Also humbling that you said nice things about me. But I want to say that, Viola Vogo also share a lot of the credit.
mhm.
Maybe we did not discover like 40 some catch.
Oh, sorry. I-
Well, so the question that I have are two questions. The first one is that the vinculum is part of the, I guess, some people model it included as the clutch model.
Mhm,
So,
mhm.
mechanical sensing, migration,
Yep,
adhesion, the dominant model is the clutch model, which
yep,
they take into account the multiple linkages of that for adhesion, the
yep.
transmit force, etc. Right? I
Mhm,
wonder, you knock it out and you saw the migration show that.
mhm,
I
mhm.
wonder whether or not, you can say something about the impact of that finding into the clutch model. There was
So,
a new Nature Physics paper
mhm.
that, refined the model. I have not read the paper. I don't know whether or not it's included in there, but I'm pretty sure that they didn't have cats farm or vinculate.
Yeah. So, I think I've seen that paper. We also have our own catch bond model or our own clutch model that I didn't talk about. And so when we formulated it, we did go into kind of the single molecule biophysics literature and get the rates of as many as we could of the force sensitive off rates. We had talon and we had vinculin. So we had kind of a branching model, and we did it for both focal adhesions and at cell-cell contacts. So in that context of that model, you can see what happens when you remove vinculin. And so I know a lot of people don't like, there seems to be resistance in the people who do most of the clutch models to put in the catch bonds, which I don't fully understand. I think you can get many of the physics that you want and find in this string with just the slip bond. But we were able to kind of find a reason. We were mainly interested in cell-cell contacts, and we were interested in kind of friction between cells. And we were able to show that basically vinculin's ability to form a catch bond is very important in determining the friction between cells, which determines how they collectively migrate. So so
That's a great answer.
yeah,
It's what
yeah,
I would have expected.
yeah.
The question is quick. I know you have set up your shop of doing all of this. I wonder whether or not we can outsourcing something.
What do you mean? Oh yeah. Wait, sorry, what do you mean by out?
We want to have a sensor, but you are the best to make those, so it's not easier to outsource.
So yeah, no, well, the problem is they're easy to make, but there's about 2 years of controls you have to do because we're inserting a gigantic two proteins inside one protein. And so it's really kind of getting over that hump. And so there have been some kind of recent advances in protein engineering for other purposes about putting things inside proteins and having the protein be functional. And so I haven't had a chance to dive into that literature yet, but with the kind of modules that we have that can have any given force sensitivity, if you can start to have confidence that you can put the module in a place in your protein and have it be functional without doing 2 years of controls, I think that's probably the key thing to get it in other people's hands. And other than, and I know Khalid had said this as well, being good at quantitative microscopy and sufficient at the analysis are not skills that everyone has, although they can definitely learn. But that's usually the second bottleneck, is just being able to do all the image analysis to do the stuff right.
Thanks.
All right, Viola.
Yes, thank you, Brenton, for spending the time today in the talk to put a lot of the mechano aspects in quantitative terms. One question I have for you: you do a lot of calibrations and solution, but
Mhm,
then certainly in the environments where you study the proteins, you have highly
mhm,
crowded conditions.
mhm,
And
mhm,
particularly the cool length already depends on
mhm.
crowding in
Yep. Mhm,
significant ways. So how do you deal with that in
mhm.
how you want to go forward?
So two answers. One, so you're right, we do many measurements of the persistence length in solution. We also often can do it inside cells. So when we measure the persistence length with the fluorescent proteins, we can do it in both. And to your point, it's often different in a cell than it is in a cuvette. And so that's kind of how we get around that, is we do think it's important to use these persistence length measurements from inside a cellular environment. So to your second point about crowding, I'll answer in two ways. The first one is there are crowding sensors that don't look all that different from some of these tension sensors. There seems that, I think if you noticed when I was making, I don't know if you noticed when I was making polypeptides, I stopped at about 80 or 100 number of amino acids. That is about the length that you can have before you start seeing some of the crowding phenomenon. And then the other one about how we handle the crowding and changing the Kuhn length. So yes, there's a question about how are we changing the persistence length of this thing at all? It's an unstructured polypeptide. I do think one of those could be, I think there is an effect of the solvent type, if you will, between the in vitro and the in cellulo environment. What we think we're doing with these new linkers, at least, is there's a lot of polyproline there. And so this is something I had to learn. There's multiple types of helices. You probably know this, but there's also a polyproline 2 helice. And so it's not super stable. So what we think is happening when we're starting to mix in a more of this proline, we gotta get like weak helical content, not like something you'd see in a crystal structure, but little helices that are there for a while, then go away and then form. So it's not truly unstructured anymore, but it's not really structured either. Does that? And so now you can kind of think of that as you have little segments that are essentially these little helical domains that are there for a little bit. And that's how we're changing the Kuhn length, by we changing the composition. That is our hand wavy working model at this time, but that's how we're thinking about it.
Thank you.
And Brent, just to follow on that, so you're able to increase the persistence length up to a nanometer, but can you go lower than that 0.5 nanometer persistence length for the
Yeah. No, so we have not, just if you do GGS, that's our softest one. If you look in solution in vitro, it'll go down to 0.3 or 0.2. I didn't do this, but the flagella one, the flagelliform one, the GPGGA, if you look in solution, that one's 0.7. So that's why we did this entire idea. Then we're like, wait, we should put them inside a cell, and we did, and they both became 0.5. And we're like, what is happening? And so to answer your question more precisely, inside cells, we have not been able to go below 0.5 nanometers.
And that may be because of that crowding effect.
I don't know. I don't know if I want to call it crowding per se. Maybe solvent or the silicity. I don't think at these lengths the sensors being smushed together. Is that we in some other work, like, yes, I called this a tension sensor, and that is how it's designed and be calibrated in. But these polypeptides do kind of like act like springs with a rest length. And so in certain contexts, we think, well, no, Val Weaver and Matt Pazak had a paper on this where they put one in glycocalyx, and they can see it can be compressed. So that's not calibrated in that mode, but you can definitely push on the thing and shove the spring together and make the fret go up.
Yeah, no, I know that paper. That's great. All right. Well, it's 15 minutes past the hour.
Yeah.
We're kind of let's thank Brent one more time for the great seminar and for answering all these questions.
Yes. Thank you all for staying late to ask questions. I really appreciate it.